So good afternoon students. Today we are going to discuss about principles of inheritance. And variation. Principles of inheritance and variation. So generally we see the every species gives the birth of same species. For example, elephant to elephant. Elephant gives always birth to the elephant. As well, cat always birth to the cat. Rat always birth to the rat. As well, human beings always birth to the human beings. So this is because of inheritance. What is inheritance? Inheritance is the process by which parental characters are passed. Parental characters are passed to progeny. Is called inheritance. Inheritance is the process by which parental characters are passed to progeny, passed on to the progeny, is called inheritance. But we see the progeny is always always similar to the parent, always not exactly to the parent. So there is something difference to the parents as well, something among to themselves. The progeny, suppose even siblings also, they show differ. That is because of variation. Variation, inheritance and variation. Variation means, variation is the degree. Variation is the degree by which progeny differs to parent. Progeny differ to parent. So this is called variation. Inheritance and variation both constitute the genetics, the study of genetics. Genetics is the study of inheritance and variation is called genetics. Inheritance, something passed from parent to progeny. That is why every species gives birth to the same species. Every species, for example, whether it is plant or whether it is animal, every species gives birth to the same species. This is because of inheritance. As well, the progeny is not always similar to parent, not exactly. There is something difference of progeny to the parent. Progeny to the parent, that is called variation. So variation is the degree by which progeny differs to parent, is called variation. The study of those inheritance and variation is called genetics. So father of genetics, is Gregor Johann Mendel. He conducted hybridization experiments on pea plants. Conducted hybridization experiments on garden pea plants. Hybridization experiments. So genetics is the study of inheritance and variation. Inheritance is the process by which parental characters are passed to progeny. That is why every species gives birth to the same species. Variation is the degree by which progeny differs to the parent. So the study of both inheritance and variation is called genetics. So father of genetics is Gregor Johann Mendel. He conducted hybridization experiments on pea plants. So in inheritance, something is passed to parent to progeny. What is something? Something is passed to parent to progeny. Right? What is something? Mendel said that the factors. Factors which are passed parent to, which carry the characters from parent to progeny, those are called factors. Now these are called genes. Now the factors are called genes. Initially Mendel called them factors. Mendel did not see the factors. Mendel could not provide the physical existence of factors. But they imagined something is there in the parent that is passed to the progeny. The something is factors as well. Now those are called genes. What is gene? Gene is the segment of DNA coding for RNA. That may be mRNA, that may be tRNA, that may be rRNA. RNA nothing but mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. So, gene is the segment of DNA coding for RNA. That may be mRNA, tRNA, rRNA. That is called gene. The segment of DNA. Segment of DNA is called gene. Where is the location of DNA? So this is the DNA. So it is located in chromosome. It is located in chromosome. This is the DNA. Every segment nothing but a gene. There are thousands of genes are present in 46 chromosomes of human being. Right? It is one of the gene. Gene nothing but a segment of DNA that is transcribed to mRNA, that is translated to a protein. Protein nothing but protein may be enzyme, protein may be hormone, protein may be receptor, protein may be collagen, protein may be antibodies. So these are all proteins. Proteins converted into everything. Protein may be melanin. So, which are all phenotypic characters. Phenotypic characters. Phenotypic characters nothing but the physical appearance of characters. The physical appearance nothing but phenotype. Physical appearance of an organism. Physical appearance of an organism is called phenotype. Only the phenotype we can see in any organism. What we can see in any organism, that is phenotype. It is expressed by the genotype. What is genotype? Nothing but genetic makeup. Genetic makeup, genotype. Genotype is genetic makeup of an organism. Genetic makeup of an organism. So this is present in chromosome within DNA, nothing but a segment of DNA. A segment of DNA. That is gene. What is the location of gene? Location of gene is locus. It is another chromosome. So it is DNA. These two are homologous chromosomes. Homologous. Homologous chromosomes. What are homologous chromosomes? Homologous chromosome is the two chromosomes that carry the same genes. That carry the same genes. We have 46 chromosomes, in which 23 as well 23. These are the two genomes. It is one genome. It is another genome. One genome and another genome. There are two genomes. These two genomes comes to progeny from the two parents. Father gives one progeny, one genome, nothing but haploid set of chromosomes. Haploid set of chromosomes. Haploid set 23 chromosomes. Haploid set 23 chromosomes. It is totally constitute a genome. It is totally constitute a genome. Haploid set of chromosomes is called genome. Right, there are two genomes in which one chromosome. Those are homologous. Second chromosome of father, second chromosome of mother. Homologous chromosomes. Third chromosome of father, third chromosome of mother. Homologous chromosomes. This is totally like 23. This is totally one genome. Nothing but haploid set of chromosome. It is one genome, haploid set of chromosomes, right? For example, it is first chromosome. It is also first chromosome. First chromosome of father present in the progeny. First chromosome of mother present in the progeny. And each individual constitutes of same kind of chromosomes. 1, 1, 2, 2, 3, 3, 4, 4 like that. So these are homologous chromosomes. So this is also the locus. It is totally locus of gene. A gene. Those are same genes that express the same characters. But each gene has two alleles. Each gene has two alleles. Genome, chromosome, gene. Each gene has two alleles. Alleles. What are alleles? Alleles nothing but alternative forms of a gene. Alternative forms of a gene is called allele. Two different forms of a gene. Those are called alleles, which are present on a locus. Those are alleles. Right. Alleles nothing but alternative forms of a gene, which may be, for example, so capital T are there. These are the two alleles of a same gene. These are same, so they are called homozygous. Homozygous. Nothing but pure. Pure nothing but genotypically, phenotypically they are similar. What is the genotype? As well the same phenotype is there. They are pure. Two alleles are same. Homozygous, capital T, small t. Heterozygous, heterozygous, nothing but hybrid. So homozygous nothing but pure. Heterozygous nothing but hybrid. As well small t also homozygous. It is also pure. Now what is the difference between two pures? Homozygous pure, heterozygous pure, homozygous pure, right? But it is the dominant one. Dominant. It is recessive. Pure recessive, pure dominant. Pure recessive, pure dominance. So these are all alleles. So same gene, two different forms. Two different forms but the same gene, that is the height. Gene of height constitute of two alleles, both are same, that is called homozygous, nothing but pure. As well heterozygous, nothing but hybrid. As well homozygous, also pure, but it is recessive, it is dominant. Dominant and recessive. Right. So here, what is the duty of gene? Duty of gene nothing but that is transmitted from parent to progeny. Earlier it was called factors by Mendel. That is transmitted from parent to progeny as well expressed to form phenotypic characters. Every phenotypic character is expressed by the gene that is present in DNA. That is present in DNA. Right. So Mendel conducted hybridization experiments on garden pea plants. Mendel conducted hybridization experiments. Experiments on garden pea plant. Garden pea plants. Why Mendel selected garden pea plant? The reasons for selection of garden pea plant. First one, it is annual plant. With well-defined characters. Well-defined characters. It can be grown. It can be grown and cultivated easily. It has bisexual flowers. It has bisexual flowers having both male and female sex organs. It can be self-fertilized easily. It can be self-fertilized easily. It has short lifespan. It has short lifespan that could be generated large number of progeny. Large number of progeny. So these are the main characters. Based on that, Mendel selected the garden pea plant. First one is, it is annual plant with well defined characters. As well, it can be grown and cultivated easily. It has bisexual flowers with both male and female sex organs. It can be self-fertilized easily. It has short lifespan, so that could produce large number of progeny. Right. So Mendel conducted hybridization experiments. So Mendel selected the characters on pea plant, that were seven characters selected by Mendel. Mendel selected 7 characters, as well 14 traits. Each character has 2 traits. First one is plant height. So it is the character plant height or stem height. Tall, dwarf. So these are the traits. So these are all alternative forms. Traits. Each one has one allele. Tall has one allele and dwarf has one allele. That is, the plant height is the character. Second one, flower position. Axial and terminal. Third one, flower color. Violet, white. These are contrasting traits. Contrasting nothing but opposite traits. Tall is opposite to dwarf. Axial is opposite to terminal. So these are the dominant traits. These are the recessive traits. Traits are 2 types. One is dominant, another one is recessive. So they are called contrasting traits. Traits contrasting. Contrasting traits. Opposite traits. Nothing but one has dominant allele, another one has recessive allele, right? Fourth one, pod shape, inflated is dominant and constricted is recessive. Fifth one, pod color, green is dominant and yellow is recessive. Seed shape, round is dominant and wrinkled is recessive. Next, the seventh character, seed color. Yellow is dominant and green is recessive. So these are the 7 characters selected by Mendel on garden pea plant. Each character has 2 contrasting traits. Every character has contrasting traits in which one is dominant. It is dominant trait. It is recessive trait. Dominant and recessive trait. Plant height, tall is the dominant, dwarf is the recessive, and flower position, axial is the dominant, terminal is the recessive, and flower color, violet is the dominant, white is the recessive. Pod shape, inflated is the dominant, constricted is the recessive. Pod color, green is dominant, yellow is recessive, and seed shape, round is the dominant, wrinkled is the recessive. Seed color, yellow is the dominant and green is the recessive. So these are the 7 characters selected by Mendel. Right. Mendel conducted hybridization experiments. Hybridization experiments. Conducted hybridization experiment in the year 1856 to 63. Mendel conducted hybridization experiments and published. Conducted experiments, and in the 1865 he published his work in the title of in the name of title experiments on plant hybrids. Experiments on plant hybrids. Experiments on plant hybrids. So Mendel conducted hybridization experiments in the year between 1856 to 63, and he published in the year 1865 in the name of experiments on plant hybrids. In the name of experiments on plant hybrids. So he was succeeded because of the first one, statistical analysis and mathematical logic. Or applied to statistical analysis, mathematical logic applied to solving the problems in biology. Solving the problems in biology. Second one, large sampling size. You take large sample that gave greater credibility to the data he collected. Next one, the results. is results only based on experiments not on unsubstantiated ideas. So these are the three major points that gave greater credibility to the Mendel. So the first one is statistical analysis and mathematical logic. He used statistical analysis and mathematical logic for the problems in biology. That is the major first one. And then large sampling size. So he selected the large sampling size that gave greater credibility to the data that he collected. Next, the third one is results. His results not based on unsubstantiated ideas, only based on the experimental results. Only based on the experimental results, he concluded. So that is why Mendel was succeeded in conducting of hybridization experiments. So right, the first experiment Mendel conducted, the first experiment is monohybrid cross. Monohybrid cross. Inheritance of one gene. Inheritance of single gene is called monohybrid cross. Mendel conducted 7 monohybrid crosses. Mendel conducted 7 monohybrid crosses. Because of Mendel selected 7 characters. On each character he conducted one monohybrid cross. For example, stem height or plant height, Mendel conducted one monohybrid cross in between tall and dwarf. As well, flower position, axial and terminal. In between these two, Mendel conducted monohybrid cross. Like that, all characters, Mendel conducted monohybrid crosses. So, Mendel conducted 7 monohybrid crosses. So, what is monohybrid cross? A cross between two individuals differ in one character. A cross between two individuals differ in one character is called monohybrid cross. Monohybrid cross. So remaining characters, they are same. They are homozygous except the character that is heterozygous. Only one character is heterozygous. Remaining total characters are homozygous. In between those two plants, Mendel conducted the hybridization experiment, monohybrid cross. So for example, tall and dwarf. So these are the parents. Parents always should be pure. Mendel conducted the female parent as capital T, nothing but tall plant, homozygous tall, right, cross between dwarf, male dwarf, homozygous dwarf, homozygous tall, homozygous dwarf, homozygous. Already we know what is homozygous, nothing but pure. It is also pure, homozygous. If there are heterozygous, how they convert into homozygous? The heterozygous plant is converted into homozygous by continuous self-pollination. This is very, very important. Continuous self-pollination leads to continuous self-pollination result in homozygosity. Continuous self-pollination result in homozygosity. Nothing but pure. Homozygous nothing but pure. Always Mendel selected pure plants for his experiment that give correct results. Otherwise, the result will be deviated. So always Mendel selected the parents as homozygous. If the homozygous are not present, the heterozygous to be converted into homozygous by continuous self-pollination. So continuous self-pollination result in homozygosity. For example, if there is heterozygous plant, then we self-pollinated by same heterozygous plant. That result in, like, for example, two gametes are there, capital T, small t, as well capital T, small t, two gametes are there, capital T, small, capital T, capital T, small t, capital T, small t, small t, small t. So automatically homozygous tall and homozygous dwarf appeared. As well, these are all next generation. If we continue self-pollination, next generation, we can get a number of stable straight inheritance. We can see the stable straight inheritance. The continuously for several generations show the homozygosity is called stable trait inheritance. That is because of continuous self-pollination. Continuous self-pollination. Because of continuous self-pollination, we can see the stable trait inheritance. Nothing but homozygosity for several generations. So automatically we can select the pure plants as parents. Pure plants as parents, right? Parents are always pure. Parents are always pure. Parents should be homozygosity. Parents should be homozygous. If homozygous are not available, the heterozygous plant must be converted into homozygous by continuous self-pollination. The continuous self-pollination result in homozygosity. It is very, very important. Parents are always pure. Parents are always should be pure. Right. It is one of the female parent, capital T, nothing but dominant homozygous, as well male parent, small t, as recessive parent. So automatically they form gametes. They form gametes. So right, both gametes are same. Then we take single, capital T, it gives small t. So these are the gametes. Gametes are formed by meiosis. During the meiosis, the diploid condition becomes haploid condition. So always gametes carry the only one allele. Gametes are always carry the single allele. That is why gametes are always pure. No hybrid condition is appeared in the gametes because gametes are always pure. Always they carry haploid genome, one genome. They carry one genome. So that is why they are pure. Right. These are the gametes. This is the parent. Right. The first generation, if we made cross between these two, automatically right, capital T, small t, we can get the hybrid. And a heterozygous plant, F1 generation, nothing but F1 generation, first filial generation, the first progeny generation. F1 generation is always hybrid. Always. It is very, very important. Always hybrid. One of the parental character did not appear in F1 generation. One of the parental character, nothing but recessive parental character, did not appear in F1 generation. Only one of the parental character appeared in F1 generation. That is dominant parental character. Dominant parental character. What is dominant? What is recessive? Dominant. Dominant means the character that is appeared in both homozygous and heterozygous condition. The character that is appeared. The character that appeared in both homo and heterozygous condition. The character that appeared in both homo and heterozygous condition is called dominant. So for example, capital T, capital C, only we can see the tall. Capital T, small t, we can see the tall. So capital T is the genotype, then we see the phenotype tall. The capital T expressed as tall. Capital T, small t expressed also tall, because here dominant, capital T is the dominant over the small t. So only the dominant character appeared in F1 generation in hybrid condition. Only dominant character appeared in a hybrid condition. That is why in homozygous condition and heterozygous condition, in both conditions, only the character that is dominant appeared. The dominant character means that appeared in both homozygous and heterozygous condition is called dominant. Now what about recessive? Recessive character. The character that appears only in homozygous condition. Only in homozygous condition is called recessive. Example, small t, small t. If the two alleles, those are recessive, then the character appears. Otherwise, that is suppressed. If one dominant allele and one recessive allele is there, automatically the dominant allele dominates over the recessive allele. Automatically, only dominant allele expressed phenotypes. Then we can see the only dominantal character, right? Only this condition we can see the dwarf. These two conditions we can see the tall, and this condition we can see the dwarf. Recessive ante only the character that appears in homozygous condition, that is called recessive. The character that appears in homozygous and heterozygous condition is called dominant. Dominant and recessive. These are the two words very, very important. Right, the F1 progeny. The F1 progeny is always hybrid, always heterozygous. Always heterozygous, right. Mendel conducted self-cross in F1 generation. Mendel conducted self-cross in F1 generation, right. Capital T, small t. Then automatically they give the gametes. This gives capital T as well small t. This gives capital T as well small t. So these are the gametes of, right, gametes. So first filial generation is always dominant. First filial generation is always heterozygous. Always heterozygous because of one dominant allele is there, one recessive allele is there. Automatically dominant allele dominates the recessive and appears. Right. So these are the 4 gametes. Right. Reginald C. Punnett, R.C. Punnett, R.C. Punnett, British genetist, created a Punnett square, right. So it is the Punnett square. It is a graphical representation. British genetist discovered the Punnett square. It is a graphical representation. So on left hand top border we place the gametes capital T, small t. The probability of gametes are shown in the left hand top border, and in the boxes possible genotypes appear in the boxes. Capital T, right, it is the tall plant. Capital T, small t, it is the tall plant. Capital T, small t, it is also tall plant. Small t, it is dwarf plant. Right, we can see 3 tall. We can see nothing but that is phenotype. Only we can see the character, that is phenotype. Genotype we can't see. By watching the phenotype we can imagine the genotype. Right, tall, dwarf. How many tall? There are 3 tall. 3 tall plants. 3 tall and 1 dwarf plant. So it is the phenotypic ratio of monohybrid cross. Phenotypic ratio of monohybrid cross is 3:1. 3 are tall plants, 1 is dwarf plant. 3:1 is the phenotypic ratio. What about the genotypic ratio? Genotypes: pure homozygous tall 1, heterozygous tall 2, homozygous dwarf 1. So this is the genotypic ratio. Of monohybrid cross, 1:2:1. The character that did not appear in F1 generation appeared in F2 generation. The character that is dwarf, it is one of the parental character that did not appear in F1 generation, automatically appeared in F2 generation. F2 generation we can see the tall as well dwarf. This dwarf character is absent in F1 generation. F1 generation 100% heterozygous. F1 generation 100% heterozygous. F2 generation there are 3:1 phenotypic ratio, in which 3 tall plants are there. 1 is pure tall, remaining 2 are hybrid tall, and 1 is dwarf. Capital D, small t, dwarf character. The dwarf character is absent in F1 but appeared in F2. F2 generation. Right. For example, one of the neat question is there. How many What is the ratio of dominant and recessive alleles in the F2 monohybrid ratio? For example, right, here is the question. Ratio of dominant and recessive alleles in F2 monohybrid cross. That is the question. Options 1, 1:1, options 2, 1:2, option 3, 2:1, option 4, 3:1. So it is the question. What is the answer? For example, capital T, capital T, capital T, small t, capital T, small t, small t, small t is there. So it is 1, this is 2, it is 1 is the genotypic ratio. 3:1 is the phenotypic ratio. It is genotype, it is phenotype, phenotypic ratio, genotypic ratio, 3:1. So if the question is ratio of dominant and recessive alleles in the F2 monohybrid class. Dominant and recessive alleles, 1 dominant, 2 dominant, 3 dominant, 4 dominant. Recessive, 1 recessive, 2 recessive, 3 recessive, 4 recessive. So 4:4. Nothing but 1:1. So answer is first one. That is the ratio of dominant and recessive alleles in F2 monohybrid class. What is the ratio of F1? Dominant and recessive alleles of F1 also same answer. Capital T, small t, that is hybrid. Automatically 1:1 is there. 1:1 is there. Right. So it is the monohybrid class. Based on the monohybrid class, Mendel proposed 2 laws. Two laws of inheritance. First one is law of dominance. Law of dominance. Law of dominance means if there are 2 alleles in which one is dominant, another one is recessive, automatically the dominant one appears as phenotype. Appears as phenotype. So generally it is the dominant character. Law of dominance, in which Mendel proposed characters are controlled by discrete units called factors. Factors. Something is transmitted from parent to progeny, proposed by Mendel. The something is factor. Now that is called gene. So right, factors. Factors occurs in pairs. In a dissimilar pair of factor, one dominates over the other. one dominates over the other. Right. Law of dominance explains characters are controlled by discrete unit called factors. Factors occurs in phase. In a dissimilar pair of factors, one dominates over the other. Right. Also, Mendel explains expression of only one of the parental character in F1. Expression of only one of the parental character in F1. Both the parental characters appeared in F2. The ratio of F2 is is 3:1 of dominant and recessive. Dominant and recessive phenotypes. Dominant and recessive phenotypes. So these are all explained by Mendel. That is called law of dominance. Law of dominance explains characters are controlled by discrete unit called factors. Factors occurs in phase. In a dissimilar pair of factors, one dominates over the other. As well, Mendel explains expression of only one of the parental character in F1 generation. Already we have seen the F1 is always hybrid, that is one of the parental character. As well, both the parental characters appeared in F2. As well, we have seen 3:1 ratio. 3 is the dominantal parental characters, 1 is the recessive parental characters, both are appeared in F2. As well, the ratio of F2 phenotype is 3:1. F2 phenotypic ratio is 3:1. So, this is the first law explained by Mendel, that is called law of dominance. Law of dominance means whenever the two alleles are there, those are in heterozygous condition, automatically this law is applied. So capital T dominates over the small t. Dominant character dominates over the recessive character. Automatically we can see the phenotype only the dominant. So that is the law of dominance. Law of dominance, right? And the second law. Law of segregation. Law of segregation. Segregation means separation. Whenever a pair of alleles present in heterozygous condition, alleles present in heterozygous condition, they do not show any blending. At the time of gamete formation, The two alleles segregate without blending. Two alleles segregate independent of each other. Independent of each other. It is called law of segregation, also called law of purity of gametes. Law of purity of gametes. Already we have said gametes are always pure. Gametes are always pure. For example, two alleles in heterozygous condition. Two alleles present in heterozygous condition, right? It is hybrid. These two alleles present in heterozygous condition. They do not show any blending. Even though they are united, they do not show any blending. At the time of gamete formation, they segregate without blending. They segregate without blending. This allele enters into one gamete. This allele enters into another gamete. Automatically, they are transmitted to progeny, nothing but gamete, without blending. This is called purity of gametes. Law of segregation. It is universal phenomena. Law of segregation is universal phenomena. Law of segregation is universal phenomena. It is applied to monohybrid, monohybrid, dihybrid, as well incomplete dominance, codominance, trihybrid. So every cross follows the law of segregation. So that is why law of segregation is universal phenomena. It is also called purity of gametes. Whenever the two alleles are a pair in heterozygous condition, the two alleles do not show any blending. As well, during the formation of gamete, at the time of formation of gametes, they segregate independent to each other without any blending. This is called the law of segregation, law of purity of gametes. So, it is totally the monohybrid cross. Monohybrid cross nothing but a cross made between two individuals different single character. Remaining all character they are homozygous except the one character, that is heterozygous. In between two plants with difference of single character is called monohybrid cross. Right, monohybrid cross, right. Mendel conducted 7 types of monohybrid crosses based on 7 characters. So it is very, very important. We must understand the concept of what is the concept inside the genetics, what is the concept inside the inheritance, what is the concept inside the variation, how inheritance is applied, how variation is appeared. So every species generates its own species, that is because of inheritance. Automatically always an elephant gives birth to baby elephant, as well human being always birth to human being, but the progeny is not similar to parent, not exact copies to parent. There is something difference in among parent to progeny as well among siblings in between among the progeny. That is called variation. These both regulate the transmission of characters. These both regulate the transmission of characters. It is very, very important concept. As well, dihybrid cross also very, very important. Total this lesson is very, very important. So, okay. Thank you, students. If you got any doubt, there is a number scrolling in the screen. You must call on it. So test cross and back cross. Test cross as well. Back cross. What is test cross? What is back cross? What is the importance of test cross? Test cross. For example, capital T is the parent to small t is the parent. Automatically we can gamete, right? We can get. It is the F1 generation. It is always hybrid 100%, right? The F2 generation is self-feed, right? Automatically we can get to capital T, small t. Capital T, small t, capital T, small t, small t, small t. So it is the F2 generation. So we can get 3 dominant, 1 recessive. We can see the 3 dominant, but we don't know what is the genotype of those 3. What is the genotype of those 3? It is F1, it is F2. So we have 3 plants dominant, tall, and 1 plant is dwarf. But 3 plants we can see, that is phenotypic. But we don't know what is the genotype of the plants. Automatically, to know the genotype, test cross is used to identify the genotype of F2 progeny according to NCERT. F2 progeny or any unknown dominant plant. Unknown dominant plant. We know the genotype of the dominant plant, whether it is homozygous or whether it is heterozygous. That is done by test cross. So automatically test cross means hybrid with recessive parent. Hybrid with లేదా F1 లేదా F2 progeny into recessive parent. Why not F1? Why we take F2? F1 is always hybrid, we know that is hybrid, though we don't need a test. But F2 there are 3 plants, 3 are dominant, but in which we don't know which is homozygous, which is heterozygous. That is why we need a test. So the test class is used to determine the genotype of the dominant parent, right? F2 progeny with recessive. For example, capital T, small t is there, small t is there. So capital T 1 gamete, small t 1 gamete, as well small t 1 gamete. Automatically we can get capital T, small t, small t, small t. This is the monohybrid test cross ratio. Monohybrid test cross ratio. Monohybrid test cross ratio is 1:1. So it is the hybrid plant with recessive parent. Automatically we can get 1:1. If we get capital T into small t, this one, capital T into small t, automatically we can get capital T, small t, 100%. Tall plants are there. So it is homozygous plant. We can determine by test cross. These are heterozygous plants we can determine by test cross. So it is the importance of test cross. Back cross means always the progeny is crossed with the parent, either of the parent. Always a progeny is crossed with either of the parent is called back cross. Recessive parent is called test cross. Test cross is also a back cross. Because the F2 progeny, unknown, nothing but those are dominant, but we don't know the genotype. The unknown parent is crossed with the F1 recessive parent is called test cross. Right. Back cross means F1 progeny is either crossed with either of the parent. F1 progeny is crossed with either of the parent is called back. Back means progeny with parent, back. But only test means F2 has dominant plants, but we don't know which is the homozygous dominant, which is the heterozygous dominant. Automatically we need the test. That is why we use the test cross to determine the homozygous or heterozygous, and we determine the genotype of the progeny. Right. Okay, students. Any doubt? Please call to the number scrolling in the screen.